And then as for the last homework assignment, it was more of like a just you guys do it kind of thing. So there's no like, we don't really have to go over it. Then as usual, if you have any questions or anything, feel free to use the chat or unmute yourself. And I will do my best to keep up with everything. Okay, can everybody see my screen? Okay, great. We go to present mode and then we can start. Okay, so today we're going to talk about the central dogma. And it's, if any of you have taken like a more advanced math class, you'll learn these things called fundamental theorem. Like there's a fundamental theorem of algebra, of geometry, of calculus, stuff like that. This is kind of the fundamental theorem of biology. Yeah, okay. So what is the central dogma? It's basically the process by which your cells create proteins. And while you wouldn't think that's super, super important, it actually involves your DNA from inside the nucleus. It turns into RNA, and then that turns into a protein. And basically, the proteins help your body run the way it does. So the two macromolecules it really involves are proteins and nucleic acids. And those two things work hand in hand to enable your body to do any of the functions it does. So yeah, there are two steps in the central dogma. You have transcription, which is the first step, and translation, which is the second. And you can see from the diagram that basically what happens is for transcription, you have your DNA, and that DNA it kind of splits apart. So we know it's a double helix shape. It'll split apart. RNA will be produced, and that RNA in translation will go and form protein. So we'll talk about exactly how that happens, but that's just a general overview. And yeah, give me 1 second. Okay. All right, so first we're going to start with transcription. So what is transcription? It is the first part of the central dogma, and it basically occurs when a copy of the DNA is made. This copy is referred to as RNA. Instead of deoxyribonucleic acid, we have ribonucleic acid. Now, this is an extravagant process that includes the separation of the DNA strands, the binding of a variety of proteins, and the final formation of a copy. There are several mechanisms in place to ensure efficient and accurate DNA transcription because the effects of wrong transcription can range from no effect to your entire body would be screwed. And what happens here allows for the second part of the central dogma, which is translation. Okay. So I have a few photos of DNA just so we can kind of go over how this works. So if you don't know already, DNA is a double helical shape. And what that means is there are two strands and they kind of intertwine. The strands have a sugar phosphate backbone, and in the middle you have your base pairs. The base pairs are the really important part. The sugar phosphate backbone is kind of what protects it and shields it. So let's see, what are some of the things labeled here? Here we have like the base pairings. So G pairs with C and A pairs with T. We know that already. And this I might not have mentioned earlier, but your DNA runs in specific directions. So you have the 3' end and the 5' end. And one strand of DNA will run from 3 to 5, and the other strand of DNA will run from 5 to 3. I did that wrong, 5 to 3. And the reason for that is the way it matches up. If it were to run the other way, then transcription wouldn't work the way it does, and it wouldn't line up correctly. Here you can see the bonds that occur between the two base pairs, as well as the bonds that occur in the sugar phosphate backbone. And yeah. Oh, something I want to point out. Maybe it'll show up in the next photo. No, it doesn't. Okay, something I want to point out is you see how these DNA strands are wound? This is one strand of DNA. After being wound like this, it can be coiled again. When it's going to be involved in transcription, the strand will be less coiled, it'll be more open and available. However, if your cell's like preparing to divide, it's not really going to be producing proteins if it's undergoing cell division, right? So your DNA will coil tightly. So Richard asks, why are there 3s and 5s? I'm not quite sure why it's called 3 and 5, but that's the way that they decided to denote the two ends. And the reason for this is, so like honestly, I feel like you call it like the apple end and the orange end. I don't think it would matter. But the reason that you have these two different orders is because the way that your DNA is structured. Hold on, let me see if I can show you guys. So if you look at these, actually, you can see this in the next image. Okay, so if you look at the way that it's set, if you see like it's kind of slanted, so you have a phosphate group and then another phosphate group here. And so imagine if this end was flipped over, it wouldn't line up the exact way that it does. And so this version is called 3 to 5 because you have the phosphate group at the top here kind of thing. And this is 5 to 3 because you have the OH at the top and the phosphate group at the bottom. But yeah, okay. And then this is why it's important for DNA replication. So this is kind of just general photo of the replication process. Right here, it's called the replication fork. And you will have replication forks in prokaryotes and eukaryotes, so bacteria and humans. But aside from that, the way that translation occurs in prokaryotes and eukaryotes is vastly different. Okay, so the way that DNA is replicated is it replicates from the 3' to the 5' end. So if you look here, DNA is generated like this. Oh no, I shouldn't have clicked it. DNA, the enzyme that unzips the DNA, I'm pretty sure it's called helicase. I actually wasn't going to get into that, but I'm pretty sure it's called helicase. There are a whole bunch of enzymes, and they all end with A's. But yes, I'm pretty sure it's called helicase. Okay, so your DNA is generated from the 3 end to the 5 end, 3' to 5'. So we'll go into this in more detail in a few, but when the enzyme binds and starts replicating the DNA, it'll always do so from the 3 end to the 5 end. And so it's a little bit awkward because on one strand that works perfectly. Like if we go back a slide, on this strand, if you were replicating, it would work perfectly. It would go from the 3' end to the 5' end, and it would just go straight. And if I'm right saying helicase, as helicase unravels the DNA in this photo, it'll just keep transcribing along here. I can annotate. It'll keep transcribing along here. So it'll go in really easily. Helicase will work this way and unravel it. However, this is called your leading strand, and this is only an issue in DNA replication because in DNA replication, you're using both sides. In RNA, you're only using one side. But anyways, your helicase moves this way and it unravels this way. So this is your leading strand, and your leading strand DNA will be synthesized across. For your lagging strand, however, when you're replicating the lagging strand, what happens here is it has to synthesize from 3 to 5. So synthesis is moving this way. And you would say, how is this possible? Because you unravel as you go. So how are you able to unravel and also synthesize in the opposite direction? Well, the way this works is synthesis occurs in fragments. These fragments are called Okazaki fragments. They were discovered by, I don't even know their first names, but it was a husband and wife. And they basically discovered that the way synthesis occurs in the lagging strand is it'll still synthesize from 3 to 5. However, it'll synthesize part of it, and then as it unravels, it'll do another part, and then as it unravels, it'll do another part. And then there is an enzyme that will bind these parts together. Okay, all right. So moving on, we have various steps to transcription. These steps include initiation, elongation, termination, and processing. There are far more steps, but for the sake of clarity, we'll just break it down into these. So yeah. Okay, so before even like the first step, or actually no, this is kind of after the first step, but you'll have a promoter, and this promoter will bind to what we call, sorry, there's stuff on my screen that's blocking this. The promoter will bind to what is called the Tata box in eukaryotes. And the Tata box is referred to as Tata because of, I guess, oh my gosh, the amino acids that are in place. So the way this works is you'll have thymine and adenine after each other for a few base pairings, and that's how that basically signals that, hey, this is the start of a gene, a specific gene to be coded. And what will happen is you'll have a promoter, which is this region. It's the promoter region of your DNA, and that is where RNA polymerase, the transcription protein, will bind. Okay, so transcription, we know, moves according to the diagram this way. So this whole section is your promoter. RNA poly will bind to the promoter, and then it'll start moving in this direction. And can somebody tell me which strand of DNA the RNA polymerase will be transcribing? Hint, check the 3' to 5'. Oh, like top or bottom strand. Anybody? Yes, it will be transcribing the bottom strand. And why is that? Because it starts with 3, exactly. Okay. Let's keep going. Oh, I thought they would disappear. All right, so here is a closer look at what's happening within the RNA polymerase, and I can guarantee to you guys that that's not what RNA polymerase looks like, but it's the easiest visual. So basically you have this enormous protein that binds to the strands, and it will start transcribing. And the way it does this is it takes amino acids that are kind of just like floating around in the nucleus, and it'll basically attach them and generate the sugar phosphate backbone. And it's called RNA polymerase because it's an enzyme. What it's catalyzing is the transcription reaction. It's catalyzing the formation of bonds between the sugar phosphate backbone and the amino acids. Okay. Oh, another thing, the strand that you're using to transcribe is called your template DNA strand, and then obviously the other strand is the RNA strand. Okay. All right. So in this diagram, it's a little bit less detailed, but you can see the promoter region is right here. And this is the Tata recognition site. I'm pretty sure this is the Tata recognition site because it comes after the first recognition site. And the reason you have two is because your cells want to ensure maximum accuracy. There is a huge chance that you'll have some random Tata region in here, but the fact that you have this recognition site and then the Tata recognition site a specific distance away, it's a lot of little factors that contribute to extreme precision and accuracy. Okay, so we see RNA polymerase binding right here, and we see the DNA starting to unravel. So this is part of initiation. It's where the DNA unwinds and RNA polymerase binds to the open complex, the open strands of DNA being the open complex. And yeah, okay. Okay, so here we have like a whole bunch of different steps, and this is really important because none of the other images showed you all of the proteins and enzymes that actually go into transcription. So you have a whole bunch of transcription factors. These transcription factors, we talked a bit last week about inhibitors and activators. Some of these transcription factors are inhibitors, some of them are activators. So basically what will happen is when transcription is supposed to start, a bunch of activators will bind to it. And when it's not going, maybe inhibitors will be bound, and then a ligand will come in and change the structure of the inhibitor, creating an activator. But basically, all these transcription factors enable RNA polymerase to bind. And there are a ton of transcription factors. It's kind of like a whole complex, with RNA polymerase being like the main enzyme. So yeah, okay. So here we can see the actual coding that occurs. And so you have RNA polymerase, it binds to the coding strand. And yeah. You'll notice it's going 3 to 5. Pretty much. Okay. So the next part is termination. So there are a lot of different ways that termination occurs. In eukaryotes, you'll have polyadenylation, which is kind of, it's also known as like the addition of a poly-A tail. And basically what happens is RNA detaches. So what we have in the image, however, is what would occur in a prokaryote. So in prokaryotes, you have two different types of termination. You have row-dependent termination and row-independent termination. So row is this factor, like a transcription factor, but it does the opposite. And what it does is it'll bind to the RNA strand, and that lets the RNA know that it's time to detach. So what will happen is you will be transcribing for a really long time. And then if it's in a eukaryote, there will be a sort of signal, just like the Tata box, but at the end. And that's how your eukaryote will know how to stop. However, in a prokaryote, that doesn't always happen. So you'll have this, sorry, excuse me. So you'll have this row factor, and row will bind to the RNA and basically let it know it's time to stop transcribing. Yeah, okay. So if we go to this image, we have row independent termination, and this occurs frequently in bacteria where the RNA will start to fold in upon itself. And that basically just, it shows that it's constructing something, which says that you kind of got to stop. But yeah, okay. So here we have processing, and processing occurs in eukaryotes, and this is after the RNA is made. So as soon as the RNA detaches from the DNA and RNA polymerase detaches, you have pre-mRNA. And pre-mRNA cannot be turned into a protein because it's got a bunch of useless material in it. So instead, we have processing. So processing begins with the addition of a 5' cap and a poly-A tail. So at the 5' end, there'll be a cap. It's kind of like a specific sequence of nucleotides that'll be added. And then at the 3' end, which is the end, you have a poly-A tail. So this is polyadenylation. And it's basically the addition of a whole bunch of adenines. True to its name, poly-A, bunch of adenines. Yeah. So the next part of processing in eukaryotes is splicing. So splicing is really, it's a very detailed process, and it could be a lecture in of itself. But basically what splicing does is it removes these exons, I lied. It removes these introns and splices together the exons. So it like splices out introns and joins exons, if that makes sense, I guess. But so introns are areas of the DNA that was transcribed that don't code for anything. They don't code for a specific protein. You'll get what I'm saying a bit later. So they need to go, because if they're in there, you're going to get really weird things. Your protein's not going to look as it's supposed to. It's like basically junk in within the DNA. Now your exons, that's the stuff that you want. That's the stuff that you need, and it'll code for a specific protein and amino acid. So you want to keep those together. So what splicing will do is a spliceosome will attach to the pre-mRNA. Spliceosomes are, you can think of them as kind of like the RNA polymerase complex. There's like so many different parts, SNRPs. It's really fun to look at. So if we have some time, I might talk about it next week. But basically, a spliceosome is this huge, intricate complex, and it'll bind to the RNA, and it'll basically cut out the introns, get rid of them, and then it'll join together the exons. And then you have your mature mRNA, and it's ready to go. Okay, so any questions thus far? Sorry about the background noise. Okay, now on to translation. So translation is the second part of the central dogma. It occurs outside of the nucleus as the first part occurred within, and it's essentially the creation of a protein. So the RNA that was created in part 1 is now translated into a protein by the work of ribosomes, and these ribosomes work with other forms of RNA to acquire amino acids, which are strung into polypeptide chains, and a protein is formed. Okay, so this is really complicated, but if we go step by step, it'll be really easy to break down. Okay, so right here, we have a very important part in translation. So that is your tRNA. So this is a different type of RNA, and this type of RNA actually folds upon itself. So I mentioned that in prokaryotes, sometimes to show the end of this is getting hard. To show the end of transcription, the RNA will fold in upon itself. So RNA is unique, and it's really interesting because of its ability to fold together and make more complex structures. One such structure is tRNA. tRNA is kind of like, you can think of it as a protein, except it's just made out of straight RNA. And it's got like a whole bunch of different parts. And what tRNA is it's this part right here. This is your tRNA. And it's got a site on the RNA, and that site will bind to an amino acid. So you'll have different tRNAs for different amino acids. They're kind of specific to each other. And at the other end of the tRNA will be an anticodon. So the anticodon matches parts of the mRNA. So I know this is really confusing, but we'll get into it a little bit more. Something that's important to mention is that instead of T, you now have U. So U is uracil, and we mentioned that adenine binds to thymine in DNA. However, in RNA, adenine binds to uracil. Okay. So what we have right here is the addition of amino acids. So DNA codes for proteins, and every 3 base pairs, so you see AUG, UAC, AUC, UCG, GAU, they group them into 3s. Every 3 bases is an amino acid. So AUC right here codes for the amino acid I don't know what this is, but ILE. And right here, I can annotate. So right here is your, apparently I can't annotate. Sorry guys, give me a sec. Okay, so right here is your tRNA. And in the other photo, we know this is the anticodon. It will match to part of the mRNA. And right here is your amino acid. So somewhere else in the cell, your tRNA will go off and search for the amino acid that it's supposed to bind to. It'll bind to it, and then it'll come back. And because it matches, then you know that this is the amino acid that's supposed to be added to this gigantic polypeptide chain. And you can see that as all of these right here. I know what this is. This is serine, and serine binds to UCG. So the anticodon AGC, A binds to U, C binds to G, and G binds to C, right? So that's how we know that serine belongs there, and it'll come, it'll bind, and then the ribosome will catalyze the reaction that joins all of these amino acids together. So this is right here is a codon. It's basically a group of 3 amino acids in an mRNA. And let's keep going. Okay, so codons. Codons are basically groups of 3 amino acids that are found in mRNA. And you have codons that actually code for amino acids, and you have codons that are start codons and stop codons. So right here is MET, M-E-T. MET is the result of a start codon. So usually there are multiple start codons. AUG is the most common in eukaryotes. So pretty much any polypeptide chain, and then thus any protein, will always begin with the amino acid MET. So there are a whole bunch of different combinations that you can get out of these three different ways that you can match the base pairs. I think there's like 64. I could be wrong. And what happens is, 4 base pairs, and you can organize them in 3 ways. Yes, 64, exactly. So there's 64 different ways that you can pair them. However, there aren't 64 amino acids. There's fewer amino acids, and that's where redundancy comes in. Kind of irrelevant right now. But anyways, you have start codons and you have stop codons. So stop codons, basically your ribosome will know that, hey, nothing's supposed to bind there, and we'll all be good to go. That's the end of the protein. This right here is your N terminus, it's the start. C terminus is the end. And yeah. Okay. So here is what I was talking about. There are 64, 1, 2, 3, 4, 1, 2, 3, 4. There are 64 different ways to organize these 4 letters. And this table basically shows you all the different ways. So your first letter can start with G, A, C, or U. Your second letter can either be U, C, A, or G. And then your third letter, all of them are over here. So I don't like this table. I much prefer those circular charts. If you look it up, chances are a circular chart will be the first that comes up. But what's useful about this is I can quickly tell you. So UAA and UAG are stop codons, and UGA is a stop codon. AUG is the most common start codon. In prokaryotes, it's always different, but that's kind of irrelevant. And you can see here. So all of these, you know what? We're going to do one that I know. All of these code for serine, right? So if you see any of these combinations in mRNA, then your tRNA will know that the tRNA has to be the serine tRNA. Okay. All right, so what actually goes on in the ribosome? So you have a polypeptide chain that's already happening, and here's the tRNA. So there are 3 sites in your ribosome. For the life of me, I can't remember which site is which, but I think it's A, P, and E sites or something like that. But basically, this is the first site, this is the second site, and this is the third site. So what happens in the first site is your tRNA with the corresponding amino acid will bind and match up. So this is kind of like the matching step because this is the exposed codon. Then after matching, it'll kind of like shift down. It's like an assembly line, and the ribosome will catalyze the bond. And then after that is done, your UAG, which is now free, this tRNA, will move on to the exit site and it'll be sent away. And this right here will move over a little bit and the bond will be a little bit more, it'll be altered, solidified. And yeah. Okay. So the differences between a eukaryotic cell and a bacterial cell, so a prokaryotic cell. In a eukaryotic cell, transcription occurs within the nucleus, processing occurs within the nucleus, and then translation occurs outside the nucleus. However, in bacteria, there is no nucleus. So you'll have transcription of the DNA, which kind of just occurs in the cell, and the translation will also occur within the cell. Something important to note is if you look at our human DNA and all eukaryotic DNA, it is in lines. However, bacterial DNA is circular. And so basically there is no starting point and stopping point. It's a circle. So we know where to start because of this replication fork that is formed. And in bacteria, it actually does the same thing. That replication fork is referred to as the origin, though, because it's a little bit more complicated. Okay, so any questions on translation and transcription? Basically, any questions in general? Give it a couple seconds. Okay, so that is all for this week. If you do have questions, feel free to stay. But yeah. Oh, also, I didn't give you guys a homework assignment because I know school is kind of starting. For me, it starts this week. I don't know about the rest of you. So yeah, just like